Hey, Bio One students, Mr. Yantz. Pardon the odd-looking outfit. It says beach day at school today for snow coming. So even though it's 5 degrees outside, we're dressing like the beach today. So hey, let's pick up with day 2 of notes in genetics. And we talked about Mendel and why he was successful in the last video. I'm going to kind of go through some of his experiments and some of his things he came up with. Some just general terms, and this is from the term quiz we had the other day that you need to know for Punnett squares. P1s or P are just your parents. F1 means first generation offspring, F2 second. And the reason we use F's is because filial is Latin for generation. Okay. So first of all, Mendel didn't do anything spectacular. He took a group of pea plants, put them in the corner of his garden, and just let them self-pollinate. And he knew what was going to happen. Generation after generation, they were going to produce the same thing. Tall produces tall, dwarf produces dwarf, green seeds produces green seeds, because they're self-pollinating. You only got one set of genetics there. And some people may ask, why go to the effort? Because this was his control group. This is where he's got his pure breeding stock from. This is where he basically had something to compare to with his other experiments. So good scientific method. You always need to have a control group, and that's what these experiments did. So he just let them do their thing, and no surprises. Okay. Then this is where the fun begins. So then he decided, you know what would happen if I cross a tall with a short? Big T, little t, little t. Now remember, Mendel didn't know anything about genes and these letters or Punnett squares. This came on after Mendel. He was just making observations. So he crossed the tall with the short. Something interesting happened in the first generation. The short disappeared. Every single offspring was tall, 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 no short. Okay? And then he said, that's interesting. Well, what is, so he just assumed a tall gene, or what we call genes now, a tall factor he called them, just basically just took over. And it did. It's dominant, right? And then an interesting thing happened, though, when he crossed the offspring together and he got F2s, which would be second generation. Now these are the grandparents. The short reappeared. So he figured out it really never disappeared. Something's going on where one gene is blocking another gene from having an expression. And he came up with a theory, as we all know today, of dominant and recessiveness, right? So he came up with 3 conclusions from that very simple experiment. Then he decided, you know what? Maybe it was this some weird thing with tall, short. Let's try green seeds with yellow. Same thing happened. Let's try wrinkled seeds with smooth seeds. Same thing happened. So he came up with 3 thoughts, okay? He said that each pair of factors, he called them, we call them genes now, control the appearance of a trait. That's a true statement, right? An organism gets these factors, obviously, from their parents. And the reason he knew that is because that short gene reappeared in the second generation, so it got contributed. It just doesn't show up very often, all right? And the big concept that we still talk about today is principle of dominance and recessiveness. One gene may, the key word there is may, mask the other or prevent it from having an effect. Okay? Since Mendel's day, we've come up with a term for that. We call those alleles, which just means alternate forms of a gene. Tall, short, green, yellow. We got one trait, but we may have multiple genes that can be expressed. Take eye color, for example. I might have green eyes, which is what I have. You might have blue, you might have brown, you might have hazel. There's multiple alleles for that one trait, which is eye color. All right? And since Mendel's day, we have figured out we're going to use a capital letter for any dominant trait and a lowercase letter, same letter, because it's one trait, the height of the plant, and we'll use a little t for short. Okay? Now, here's the big thing I want you to understand. This will never be the law of dominance and recessiveness because there's exceptions to this rule. It doesn't always happen this way. I think the misconception sometimes we get is every trait out there has a dominant recessive gene. Not true. What if, okay, and let's throw some terms out there first, okay? So we come up with these terms: homozygous and pure, interchangeable words. You'll see me use those on Punnett squares. That means organisms with identical alleles, 2 capitals or 2 lowercase. That's your 2 options. Or heterozygous hybrid, which is dissimilar alleles, a big T, little t. All right. So we have this thing called incomplete dominance and codominance. They are different, but what happens with incomplete dominance, we'll do it first. What if I got 2 genes and they're both dominant? All right, so let's say, like in snapdragons, I have a red flower and a white. They're both dominant, and if they get paired up, I get a pink. They blend. And this is how we get Dalmatian dogs and Holstein cows and speckling, or spots. What if calico cats? Another great example. What if we have 2 genes? We'll never call it the law of dominance recessiveness, because there is some exceptions. So if you look at these guys, I got a red and a white, and if I get them together, I don't have red, I don't have white, I get pink. Okay. So how does that work, and how do we set that up? So we have to do a Punnett square a little bit different. Okay. So we have to use, I like to use 2 capital letters because it's the same trait, but I don't want to use a lowercase because that means one's recessive and they're not. They are both dominant. So how we show that is a little apostrophe, a little prime notation on one of the letters. So this is red, this is white. If they get together, I get an intermediate or a blend called pink. And you can see the Punnett square. So you can see dominance and recessiveness does not always play out with all traits. Most traits it does, but with some it does not. Okay, now where this gets really, really weird, okay, is what if they just both show up? They don't blend, they both just show up. That's called codominance. Okay, same Punnett square, but the difference is if the 2 get together, I don't get a pink. In this case, I get a roan. This is this horse over here. That's where the red and white hairs grow side by side, and you get a speckling, and you get some unique looking animals out of that. So that's a different type of Punnett square, which we'll talk about and do in Punnett square quizzes and things like that. All right. Another concept, and this became a law, is called the law of segregation. So Mendel figured out, okay, these genes separate somewhere during the formation of egg and sperm cells. Okay. He didn't know meiosis. That came along later. Now we have a name for it. The process is called meiosis. And the reason he knew the law of segregation occurred, if we go back to this one, okay, so let's go back a couple slides. And we go back to this simple experiment. Remember this one. He figured out, okay, these must separate because that's the only way this short one's going to reappear in the second generation. And we know that that's what happens. The chromosomes line up at the equator during meiosis, and what do? They separate. So you're going to get the big T or the little t. Okay, so that is called the law of segregation. Okay, and it is a law. It always happens that way. And we talked about that with meiosis and mitosis. All right, and then he came up with a second law, third theory, second law, and that was called the law of independent assortment. Okay, which means there's nothing that controls what genes I get. So it's like playing a card game. You got the same deck of cards, but I can play a card game because I deal out different hands. Whether you're talking about poker or euchre or whatever. Okay. Now, and we know that that's true too. Meiosis and mitosis again, right? Now, I did put a little asterisk in here, and I do want you to understand because I have students sometimes argue and say, well, wait a minute. Why? How do you explain then if it's random? It's like flipping coins. We did that little activity with the face that we made, right? Scandinavia. Why is almost their whole population in that small country blue-eyed and blonde-haired? That defies the law of independent assortment. No, it doesn't, because guess what? The gene for blonde hair and blue eyes is on the same chromosome. It moves together, right? We're only talking about the first chromosome is not going to have any bearing on the third chromosome. Third chromosome doesn't have any bearing on the 22nd chromosome. Your 23 pairs of chromosomes line up and separate, and that's why Sometimes we look a lot like our parents. Sometimes we don't look anything like our parents. Sometimes we look like our siblings. Sometimes we don't look like our siblings. It's a shuffling of the deck. Okay. So that's the other law that Mendel came up with, the law of independent assortment. Okay. Spin the clock forward a little bit after Mendel, and we came up with this grid system called Punnett squares that we've done some stuff with already. And you see with genetics, it shows us the probability of what's the odds of us getting this trait or that trait. And again, it's just a probability. It's like horse racing. The favorite doesn't always win, right? So sometimes that happens with Punnett squares. And do not miss this on the test. The inventor of the Punnett square is R.C. Punnett. Imagine that. Don't miss that question. All right. So we do have a system where we show these laws and these principles that Mendel came up with, and we understand them more now because we got better technology. We can see things better. So Mendel was right on the money with his theories and laws, and we're still talking about them today, right? All right. So I'll talk about a little different Punnett square next set of notes, sex-linked ones, and pedigrees. So we'll kind of wrap up notes with those two concepts, and then we'll take a test. All right. So, hey, have a good day. And if you've got any questions, you know what to do. Send me an email. We'll see you. Have a good day, and have a nice sunny beach day. See you. Bye.